It's kind of strange to think about, but what if a really common medicine, something maybe you've taken before, could be perfectly fine for one person,
Hmm.
but potentially really dangerous for you?
Or think about cancer treatment. A revolutionary therapy might completely turn things around for a patient, but only if their tumor has this very specific genetic marker.
Right, it sounds almost like science fiction, doesn't it? But it's actually happening now. It's called precision medicine, and that's what we're diving into today.
It really is a fundamental shift. Instead of just reacting when things go wrong, treating symptoms after the fact, we're moving towards being proactive, predictive even.
So our mission for this deep dive is to really unpack the science behind it all, how our own unique genetics are changing everything from diagnosis to treatment.
Exactly. We'll look at the real world impact, like how your body might handle different drugs, what's happening in cancer therapy, and well, also some of the big challenges that are still out there.
And we've got some great source material to lean on, mostly from a key chapter in Essentials of Genetics, the 10th edition. It's packed with info on these breakthroughs.
Yeah, it's going to help us get a handle on this really fast moving field.
Okay, so let's start with one of the biggest areas,
Mm.
how precision medicine is changing the way our bodies respond to drugs. When we say precision medicine, what does that actually boil down to for a patient?
Well, at its heart, it's moving away from that one size fits all idea, where everyone gets the same drug at the same dose.
Right.
Instead, it groups patients based on their molecular details, their specific
Well,
genetic makeup, their biology. The idea is to guide you towards the treatment that's going to work best for you with the fewest side effects. It's tailored.
okay, that makes sense. Tailored medicine. And I hear two terms thrown around a lot. Pharmacogenomics and pharmacogenetics. Are they basically the same thing?
That's a good question. They sound similar and people do sometimes use them interchangeably, but there's a difference. Pharmacogenomics is like the bigger picture. It studies how your entire genetic makeup, your whole genome, influences your drug response. It also includes developing new drugs that are designed for specific genetic profiles from the get-go.
Ah, so it's the whole kit and caboodle of your DNA and drugs.
Pretty much. Pharmacogenetics is a bit narrower. It focuses specifically on how variations in particular genes affect how you respond to a drug. Think of it as one important piece within pharmacogenomics.
Got it. Now, why is digging into this so important? You mentioned side effects earlier, and I know that's a massive problem, even if we don't always hear about it.
Oh, it's huge. Just look at the numbers in the US alone. Every year, something like 2 million people have serious side effects from medications.
2 million. Wow.
And tragically, about 100,000 of those people actually die. Plus, you've got loads of patients who just don't get better because the drug doesn't really work for them as expected. A lot of that comes down to their genetics.
That's staggering. It really highlights why we need this.
Hmm.
So walk us through it. How does our DNA actually control how we handle a drug?
Okay, think of a drug entering your body. Your genes are like the instruction manuals for building all the little workers and machines inside your cells.
Okay, like little biological factories.
Exactly. Some workers are like delivery trucks, they carry the drug around. Others are like docking stations on the cell surface receptors the drug needs to connect with. Then you have the demolition crews enzymes that break the drug down. And finally, waste management crews to get rid of it.
Makes sense.
Now, if your instruction manual, your DNA, has variations, maybe the delivery truck is slow, or the demolition crew works overtime, or the docking station has the wrong shape, any of those things can totally change the drug's effect.
That's a great way to visualize it. Can you give an example of one of those gene families that acts like a demolition crew?
Sure. A really important one is the cytochrome P450 gene family, often just called CYP genes. These guys are heavy hitters in drug metabolism. They process a lot of different medications.
So if my CYP genes are different from yours, we could process the same drug completely differently.
Precisely. Some people have variants in these CYT genes that make them metabolize drugs really slowly. The drug hangs around too long, builds up, and you risk those overdose side effects we talked about.
Okay.
Others have variants that make them metabolize super fast. The drug gets eliminated too quickly, maybe before it even has a chance to work properly, so it's less effective.
And there's one specific gene, CYP2D6, that comes up a lot, right? It affects a surprising number of common drugs.
Yes, CYP2D6 is a big one. It's involved in breaking down something like 25% of all pharmaceutical drugs. That includes stuff like acetaminophen, codeine, certain antidepressants, beta blockers, even the cancer drug tamoxifen. And get this, there are over 70 different known variations or alleles for just this one gene.
70 variations for one gene. That's wild.
Okay.
So how does that play out? If I take codeine, for example.
It means we can actually group people based on how their CYP2D6 works. Most people, maybe 80%, are extensive metabolizers. They have the common gene versions and processed drugs as expected.
Okay, the normal group.
Right. But then about 10–15% are poor metabolizers. They have 2 copies of a low-activity gene variant. For them, a standard dose of codeine might not work well, or other drugs might build up.
Yeah, the opposite.
Then you have ultra-rapid metabolizers. These folks might have extra copies of the gene. They break down certain drugs, like codeine, really, really fast. Codeine actually needs CYP2D6 to convert it into morphine to relieve pain. So ultra-rapid metabolizers might get a dangerously high dose of morphine from a standard codeine dose, or other drugs might be cleared so fast they don't work at all.
That's a huge spectrum of responses from the same pill.
Yeah.
Are there other key gene examples that really affect common treatments?
Oh, yeah. The book mentions several, like the TPMT gene variants there affect how people handle thiopurine drugs used for leukemia. Get the dose wrong and it can be highly toxic. Or, for the blood thinner warfarin, variants in 2 genes, VKORC1 and CYP2C9, massively influence how effective it is and the risk of bleeding. These aren't just, theoretical points. They matter for patient safety every single day. Another one, CYP2C19, affects drugs like clopidogrel, an antiplatelet drug, and some antidepressants. And SLCO1B1 affects how well your body removes statins.
Wow. So, knowing this, what's the practical solution? How do we use this genetic information?
Well, the big goal is preemptive pharmacogenomic screening.
Preemptive meaning before you even prescribe the drug.
Exactly. Instead of waiting for a bad reaction or for the drug to obviously not be working, you screen the patient's relevant genes before starting treatment. Then the doctor can pick the right drug and the right dose based on that person's genetic profile right from the start.
That sounds like a much smarter approach. Is this happening routinely now?
It's starting to. Tests are available for variants in around 20 or so genes that predict reactions to maybe 100 drugs. That's still only about 18% of U.S. prescriptions, so there's a way to go.
But some places are really pushing ahead. I think I read about a program at St. Jude Children's Hospital.
Yes, PGEN4 Kids at St. Jude. They started it back in 2011. It's a clinical research program, but they offer really extensive screening.
What kind of screening?
They test kids, often when they're first diagnosed with cancer, for thousands of variants across hundreds of genes that might affect drug response. The uptake was huge, like 97% of families agreed to it.
Wow.
From just one blood sample, they look at about 2,000 variants in 230 genes, and they found that nearly 78% to be exact of the kids, had at least one genetic variant that could impact how they react to a drug.
That's incredible. And they link it to their health records.
They do. The results go into the electronic health record, and it's automatically updated if new information comes out linking a gene to a drug. The idea is it's cheaper, faster, and way more efficient in the long run than just ordering tests one by one later on.
It must prevent a lot of problems.
You'd think so. There was a study out of Vanderbilt around 2012 suggesting that nearly 400 serious drug reactions could have been avoided if doctors had had this kind of preemptive screening info. It really is a potential game changer.
Okay, so that covers optimizing existing drugs. But precision medicine is also driving the development of entirely new treatments, especially for cancer. Let's talk about precision oncology.
Right, this is a huge focus. The idea is to design drugs that specifically target the unique genetic fingerprint of a patient's tumor. We now understand that every tumor is genetically different, even tumors of the same type, like breast cancer.
So you're looking for the tumor's specific weakness, its Achilles heel.
Exactly. Find that genetic vulnerability and design a drug to hit it precisely.
And one of the poster children for this approach is Herceptin for breast cancer, isn't it?
Absolutely. Herceptin, the brand name for trastuzumab, was one of the very first successes in targeted cancer therapy. It targets the HER2 gene.
What does HER2 normally do?
Normally, the HER2 gene provides instructions for making a protein, a receptor that sits on the surface of breast cells. When certain growth factors bind to it, it tells the cell to grow and divide. It's part of normal cell regulation.
But in some cancers, it goes haywire.
Exactly. In about a quarter of invasive breast cancers, the HER2 gene gets amplified, meaning there are way too many copies of the gene. This leads to way too much HR2 protein on the cell surface. This overexpression makes the cancerous cells grow faster, spread more easily, and generally leads to a worse prognosis.
So Herceptin was designed specifically to shut that down.
Precisely. Herceptin is what's called a monoclonal antibody. Think of it like a guided missile designed to lock on to just one target, the HR2 protein on the cancer cell surface. When it binds, it blocks those growth signals, stops the cell cycle, and flags the cancer cell for destruction by the immune system.
And that's why testing is so critical. Because if the tumor doesn't have that HR2 overexpression, the drug won't have anything to target.
Absolutely essential. Doctors use tests like IHC, which stains the tumor tissue to see how much AR2 protein is there, or FISH, which counts the actual number of HR2 gene copies in the cells. If the tumor isn't HR2 positive, Herceptin won't help.
But when it is positive,
When it is, and Herceptin is added to chemotherapy, it can boost survival rates by 25 to 50%. It's made a huge difference for those patients. And it's also used now for some other cancers that overexpress HR2, like certain stomach cancers.
that's a fantastic success story. Are there other examples of these targeted drugs making a big impact?
Definitely. The source material mentions a few key ones. There's imatinib, or Gleevec, which revolutionized treatment for chronic myelogenous leukemia, CML. It targets a specific faulty enzyme, the BCR-ABL kinase, that drives the leukemia.
Kinase, that's like an on-off switch for cell growth.
Exactly. And in CML, that switch is step on, Gleevec turns it off. Another example is olaparib, or Lynparza. It's used for ovarian cancer patients who have mutations in the BRCA1 or BRCA2 genes. It blocks another enzyme called PARP, which is involved in DNA repair. Cancer cells with BRCA mutations rely heavily on PARP, so blocking it is particularly effective against them.
So these drugs can be incredibly powerful for the right patient. But it sounds like finding that right patient is still a challenge.
It is. While the successes are amazing, the reality right now is that only a relatively small percentage of cancer patients can currently be matched to an available targeted drug based on their tumor-specific mutations. One trial mentioned found only about 6.4% of patients could be matched.
Still, for that 6.4%, it can be life-changing. Which brings us to that incredible story of Dr. Lucas Wortman. It really shows the potential, even when the odds seem impossible.
Oh, the Wortman story is just remarkable. He was a physician himself, battling a very aggressive form of leukemia, ALL. He'd gone through multiple rounds of chemo, a bone marrow transplant. Nothing was working long-term. His cancer kept coming back, and things looked really bleak.
This was back in 2012.
Yes. And his colleagues at Washington University, where he worked, decided to try something radical as a last resort. They decided to sequence everything.
His entire genome.
Both his normal DNA and the DNA from his cancer cells. And not just the DNA, they also did RNA sequencing, called RNA-seq, to see which genes were actually switched on and how active they were in the cancer cells.
What did they find?
They found a bunch of mutation, as you'd expect in cancer, but nothing they had a drug for. But the RNA analysis showed something surprising. A gene called FLT3 was massively overactive, way more than normal. It coded for a kinase, one of those signaling enzymes.
Okay, so they found an overactive switch. Did they have a drug to turn it off?
Well, not one approved for leukemia. But they found a drug called sunitinib, which was already approved for kidney and gastrointestinal cancers, that happened to target the FLT3 kinase.
So they tried it.
It wasn't easy. Insurance initially refused to pay, and the drug company, Pfizer, first denied compassionate use access. But Dr. Wortman managed to buy a week's supply himself.
Yeah.
The results were stunning. Within days, his blood count started normalizing. Within 2 weeks, tests showed his bone marrow was clear of cancer. It was dramatic. Pfizer then reversed its decision, he got the drug, and was able to get a second bone marrow transplant.
That's just unbelievable.
It really is. While his long-term prognosis was still uncertain at the time the story was written, his case was pivotal. It showed the power of deep sequencing to find unexpected targets and really spurred research into FLT3 and similar pathways in other cancers. A true testament to personalized medicine.
An incredible story. Now, besides these targeted drugs that hit specific molecules, there's another huge area in precision oncology. Immunotherapy. Trying to get our own immune system to fight the cancer more effectively.
That's right. Our immune system is incredibly powerful, but cancer is tricky and often finds ways to evade it. Immunotherapy tries to tip the balance back in favor of the immune system.
So, quick refresher. What are the key parts of the immune system involved here?
Well, you have the innate system, that's the rapid first response team, things like macrophages. And then the adaptive system, which is more specialized, learns, remembers, and includes B cells making antibodies and T cells.
And for directly killing cancer cells, we're talking mostly about T cells, specifically the cytotoxic ones.
Exactly. Cytotoxic T lymphocytes, or CTLs. These are the T cells that can recognize specific markers, antigens on the surface of cancer cells, latch onto them, and kill them directly.
But you said cancer cells can evade them. How do they do that? It's like they have invisibility cloaks.
They have a few tricks up their sleeves. One big one is they can produce molecules on their surface that basically hit the brakes on T cells that try to attack. These are called checkpoint molecules.
Ah, and that's led to checkpoint inhibitor drugs.
Precisely. Drugs that block those brake signals, unleashing the T cells. Another tactic. Cancer cells can mess with the MHC molecules on their surface. MHC molecules are what cells use to display antigens, like showing their ID badge to T cells. If the badge is messed up, the T cell can't recognize the cancer cell properly.
Sneaky. What else?
Tumors can also attract regulatory T cells, or Tregs. These are actually immune cells whose normal job is to suppress immune responses to prevent autoimmunity, but in the tumor, they can shut down the cancer-fighting T cells. Other immune cells that infiltrate the tumor, like certain types of macrophages, can also dampen the T cell attack.
Okay, so cancer has multiple ways to hide or defend itself. How does immunotherapy try to overcome this?
There are several strategies, but two main ones mentioned are adoptive cell transfer, or ACT, and engineered T cells.
ACT, what's that involve?
With ACT, doctors actually take a sample of the patient's tumor, isolate the T cells that have already managed to get inside it. These are called tumor-infiltrating lymphocytes, or TILs. They then grow these TILs in the lab, selecting and expanding the ones that seem best at recognizing the cancer until they have billions of them. Then they infuse this large army of cancer-specific T cells back into the patient.
So you're basically amplifying the patient's own best soldiers.
That's the idea. The other approach, which gets a lot of attention, involves genetically engineering T cells.
Like the KR T cells we hear about.
Exactly. Chimeric antigen receptor T cells. Here, scientists take regular T cells from the patient's blood and genetically modify them in the lab. They insert genes that make the T cells produce a special receptor, the CIR, on their surface. This CIR is designed to recognize a specific antigen on the patient's cancer cells.
So you're essentially programming the T cells to hunt down the cancer.
That's a great way to put it. You create these supercharged, targeted T cells, grow billions of them, and infuse them back into the patient. They can then seek out and destroy cancer cells much more effectively. It's like giving your immune system GPS-guided missiles specifically for the cancer.
This is all incredibly sophisticated. It really feels like this whole omics revolution, genomics, proteomics, metabolomics, is the engine driving all of this progress.
Absolutely. The rapid drop in cost for sequencing, whether it's the whole genome or just the protein coding parts, the exome, has made it feasible for diagnosis in a way it never was before.
And big projects are mapping this out.
Yes, projects like the Cancer Genome Atlas have been vital. They've systematically mapped the genomic changes in thousands of tumors across many cancer types. This creates a massive database that researchers can mine to find new drug targets. And it's not just DNA anymore. We have high-throughput ways to look at all the proteins, proteomics, or all the small molecule metabolites, metabolomics in a sample. It gives a much more complete picture of what's actually going on inside the cells.
Which brings us to that fascinating case study of Dr. Michael Snyder, who basically used all these omics on himself.
Right. The Snyder study is a really cool example of what's called personal omics profiling. He tracked his own health over 14 months using a whole battery of tests.
What did that involve?
He had his whole genome sequenced, of course, but then he also repeatedly measured his transcriptome, that's all the RNA molecules, showing which genes were active. He measured his proteome, the proteins, and his metabolome, the metabolites, all tracked over time.
What did he learn from drowning himself in his own data?
Quite a lot, actually. His genome sequence showed he had certain genetic variants, SNPs, linked to increased risk for things like coronary artery disease, type 2 diabetes, even a couple of types of cancer. But the real insights came from tracking changes over time.
Like how his body responded to things.
Exactly. For instance, when he caught a respiratory virus, RSV, they could see specific networks of genes ramp up their activity and then quiet down again by looking at the RNA levels. They saw similar coordinated changes in gene expression, proteins, and metabolites when his glucose levels change.
So it's not just about the blueprint, the DNA, but how the factory is actually running day to day.
That's the key takeaway. His study really highlighted that just knowing the static DNA sequence isn't enough for truly personalized medicine. You need to monitor how genes are being expressed, how proteins and metabolites are changing in response to life events, diet, infections, everything. It's like moving from a static map to a live traffic report for your health.
This is clearly incredibly powerful stuff.
Yeah.
But it also sounds complicated and maybe expensive. What are the big hurdles we still need to overcome to make precision medicine widespread?
There are definitely significant challenges, both technical and societal. On the technical side, we still need genome sequencing and these other omics technologies to get even faster, cheaper, and more accurate.
And dealing with all that data.
Oh, absolutely. The sheer volume of data is immense. A single personal genome sequence is like the equivalent of 200 huge phone books of information. Storing that, analyzing it, interpreting what it means, it requires massive computing power, sophisticated algorithms, and large-scale studies linking genetic variants to health outcomes in diverse populations.
And it's not just genetics in isolation, right?
No, you have to integrate it all. You need to factor in environmental exposures, lifestyle choices, diet, exercise, even epigenetics changes in gene activity that aren't coded in the DNA itself. That adds layers of complexity.
Okay, the healthcare system needs to catch up.
Big time. We need robust electronic health records, EHRs, that can seamlessly store and integrate this genomic data with a patient's other health information and allow doctors to easily access and use it, cross-referencing it with the latest research. Right now, the infrastructure just isn't there in most places. Maybe only 10% of U.S. hospitals and clinics have sophisticated enough EHRs.
So lots of technical and logistical hurdles. What about the social and ethical side?
Those are arguably even tougher. There's the question of cost and resource allocation. These technologies and targeted drugs can be incredibly expensive. Is it fair to spend so much on specialized care for relatively few when basic healthcare needs aren't met for many around the world, or even within the same country?
That raises serious equity concerns.
Definitely. There's a real worry that precision medicine could worsen health disparities, becoming a luxury only the wealthy can afford. Then there's privacy. We're talking about people's most fundamental biological information. How do we protect that data in large databases and EHRs from misuse or breaches?
And educating everyone involved.
Crucial. Doctors need to be trained to understand and apply this genetic information correctly. Patients need to understand what these tests mean, and we desperately need more genetic counselors and genomic specialists to help interpret results and guide patients. As of a few years ago, there were only about 4,000 licensed genetic counselors in the entire U.S. That's nowhere near enough for widespread implementation.
It really underscores that we're at the beginning of this journey, even though the science is moving so fast.
We absolutely are. It's a revolution in progress. But the impact is already being felt. Genetic and genomic information is making drugs safer and more effective for some people, and it is helping predict disease risk.
We're shifting from generalized medicine to something truly individualized.
That's the promise. And looking forward, it's hard to imagine an area of medicine that won't be touched by precision and personalized approaches eventually, from preventing illness to diagnosing it earlier to treating it more effectively.
It's an exciting but also complex future. It leaves you wondering.
As precision medicine keeps advancing and we get better at optimizing health for individuals, what new questions does that raise? Especially about how we balance that individual focus with ensuring everyone has fair access to these potentially life-changing technologies. That's something to really think about.